Assalamu alaikum and welcome everyone to the second part of molecular biology ASCP preparation series molecular techniques. I'm Dr. Lai Abdelati and I'm really excited to share my knowledge and experiences with all of you through this series. So let's dive in together. Our focus for this season will be on the last part of the separation and detection part, probe structure. Alright, before we dive into the question section, let's briefly explore the common types of molecular probes, which will be featured in the MCQs. This includes the first one, Tagman probes, as we see in the image here. These are hydrolyzed probe used in real-time PCR, designed with the fluorescent reporter at one end and quencher at the other end. Okay, they ensure high specificity and enable quantitative analysis. The second one is the FRET probe, or fluorescent resonance energy transfer. This probe rely on energy transfer between two fluorophores to generate a detectable signal. It is widely used in study molecular interaction. Okay. The third probe here is the molecular beacons. These are hairpin-shaped probes with the fluoroform and a quencher. They fluorescence only upon binding to their target sequence, making them high specific for real-time application. The fourth probe is the scorpion probes. These are self-priming probe used in real-time PCR, combining a primer with the probe sequence in a single molecule. They provide high sensitivity and are especially useful for detecting target sequence during amplification. The last one is simple probes. As their name suggests, these are straightforward probes labeled for detecting specific nucleic acid sequence. They are often used in basic molecular diagnostic. Alright, now let's move to the question part and see the first question we have today. Alright, the first question we have today, what is the primary function of the quencher in a Tagman probe? The correct answer is to suppress fluorescent until cleavage. Okay, as we know, the quencher suppresses the fluorescence of the reporter dye until the probe is cleaved during PCR process. All right, let's move on. All right, the question here: which labeling technique is commonly used in probes? Correct answer, all of the above. Actually, probes may be labeled with the fluorescent dye, radioisotopes, or biotin to enable detection using various platforms. All right, let's move on. All right, the next question here. Which enzyme is responsible for cleaving the Tagman probe during PCR? The correct answer is tag DNA polymerase. Actually, tag DNA polymerase has a 5' to 3' exonuclease activity, as we said in the previous lectures, which cleave the Tagman probe during PCR, right? All right, what happens to the fluorescent signal when the Tagman probe is cleaved? For sure, it's going to increase. As we know, cleavage of Tagman probe, what would happen? Separate the reporter dye from the quencher, right? Resulting in increase in fluorescent. Next question. Tagman probe are commonly used in which type of PCR? Alright, Tagman props are specifically designed for real-time PCR to monitor the amplification process in real time. Alright, let's move on. Next question. Which of the following is the key advantage of using Tagman props? The correct answer is high specificity. Actually, Tagman props provide high specificity due to their sequence-specific hybridization and cleavage. Alright? The question here. What does FRET stand for in the context of molecular props? FRET stands for the fluorescent resonance energy transfer, a mechanism where energy is transferred from the donor to acceptor fluorophore. Alright? In FRET probes, what happens when the donor and acceptor fluorophores are in the close proximity? Alright, when the donor and acceptor fluorophores are in close proximity, okay, energy is transferred from the donor to acceptor, resulting in fluorescent emission from the acceptor. Alright? Okay, which of the following is the common application of FRET probes? Actually, FRET probes are commonly used to study protein-protein interactions by detecting changes in proximity between the fluorophores. Alright? Okay. What is the main advantage of using FRET probes in molecular biology? The correct answer is high sensitivity. As we know, FRET probes offer high sensitivity in detecting molecular interaction and conformational changes. Alright? Okay, next question here. Which of the following is true about the emission spectra of donor and acceptor fluorophores in FRET? Alright, the correct answer is they must overlap. Alright? Actually, the emission spectrum of the donor fluorophore must overlap with the excitation spectrum of the acceptor fluorophore for efficient energy transfer. If you found this content helpful, don't forget to like, share, and subscribe. Your support means a lot to me and help me create more valuable content for you. Thank you for being part of this journey. Let's continue. Okay, next question here. What is the simple probe in the context of molecular biology? The correct answer is a probe with a single fluorophore. Alright, let's move on. Which of the following is the common use of simple probes? The correct answer is real-time PCR. Alright, let's move on. Let's see the next question here. What is the main advantage of using simple probes? The correct answer is low cost. As you know, simple props are generally less expensive to produce compared to the more complex prop designed. Okay? Next question is, simple props are typically labeled with the which of the following? Simple props are commonly labeled with the fluorophores for fluorescent detection. Okay? Which of the following is true about the design of simple props? Okay, simple props are single-stranded oligonucleotide designed to hybridize to a specific target sequence. Okay, so the correct answer here is they are single-stranded oligonucleotides. All right, what is the characteristic structure of molecular beacon probe? The correct answer is hairpin. Okay, the molecular beacon probes had a unique hairpin structure, also called steam loop. Okay, this hairpin structure or steam loop designed for precise detection of target nucleic acids. Okay, let's move on. All right, let's see the question here. What happened to the fluorescent signal when a beacon probe hybridized to its target? The correct answer, it increases. Actually, when a beacon probe hybridizes to its target, the fluorescent signal increases. This is because the separation of the fluoroform from the quencher restore the fluorescent signal indicating the presence of target nucleic acids. Alright, let's move on. Alright, the next question. Which of the following is the key feature of beacon probes? Alright, actually beacon probes, as I said, have both a quencher and a fluoroform, which allow them to emit fluorescence upon hybridization. Okay, let's move on. Okay, next question. What is the main advantage of using beacon probes? Okay, the main advantage of using beacon probe is high specificity. Actually, these probes are designed to fluorescent only when they hybridize with their specific target sequence, all right? Which minimize false positive and enhance the accuracy of detection. All right, the next question. What is the unique feature of scorpion probes compared to the other probes? Okay, actually, scorpions probe are unique because they contain a PCR primer and a probe in one molecule. Actually, this design allows for rapid and efficient detection of the target sequence during PCR. All right, let's move on. Okay, next question. Which of the following is true about the design of scorpion probes? The correct answer, they have a quencher and reporter in one molecule as we see in the image. All right, let's move on. Next question. What happened to the fluorescent signal when a scorpion probe hybridized to its target? The correct answer, it increases. Actually, when a scorpion probe hybridizes to its target, the fluorescence signal increasing. This is because the hairpin structure of the probe opens up, separating the quencher from the reporter, which allows the reporter to emit fluorescence. Alright, let's move on. Okay, last question we have today. What is the main advantage of using scorpion probes? The correct answer is high specificity. As I said, scorpion probes provide high specificity due to their unique design, which combine a PCR primer and a probe in one molecule, ensuring precise target detection. Okay? That's all for today. I hope you enjoyed this lecture. Please don't forget to like, comment if you have any question, share and subscribe to our channel to receive all the latest lecture. Alright? I also encourage everyone taking the test to share your question with us so that everyone can benefit. That's all. Salam.